All righty. So here we are. We left off at PCR, and that's where I'm going to pick it up. Okay. There's a couple of things extra that I wanted to cover about PCR, and one of them has to do with what needs to go into the test tube for all this stuff to work. Okay. So for PCR, for polymerase chain reaction, what do we need? Well, we need a gene of interest, okay? So we need, we call it template DNA. So we need template DNA. What else do we need? Well, we need the primers, okay? If you're, again, the number of primers depends on how many locations or loci you're amplifying. It is 2 primers per location. So we say forward and reverse primers, RNA primers. Okay. What else do we need? Well, we're gonna need the DNA polymerase to synthesize it. Okay. So DNA polymerase. What else? Well, we're going to need the nucleotide triphosphates. So it's a deoxynucleotide triphosphates, specifically DGTP, DCTP, I'll get it right, DATP, and DTTP. Okay, so those are the things that need to go into a tiny PCR tube in order to amplify DNA. You need the DNA itself, you need the RNA primers, 2 per loci, you need the DNA polymerase. This is going to be DNA polymerase 3. And for those of you that already know PCR, there's always 1 or 2 out there. This comes from Thermus aquaticus. And so we say TAQ, Thermus aquaticus. This is the organism from which this DNA polymerase was harvested, Thermus aquaticus, and it stands for hot water in Latin, okay? And then your nucleotide. So that's important to know. I think that's going to be one of the things you need to know in your quiz. Another thing that you need to know is that now there's this thing called real-time PCR, okay? So what happens is that once upon a time, we still do this. We take a low PCR tube, you throw all this stuff in there, put it into a thermocycler, do a Hail Mary, go get a cup of coffee. Hope it works, okay? That is no longer the case, okay? For just a few $100s more, you can actually get something even better. And it's called real-time PCR. It says real-time PCR right there, okay? And I use my mad drawing skills from Word to see if I can explain this, okay? This blue line is my DNA. This brown line is my RNA primer. This green circle is my DNA polymerase 3, okay? So obviously we have already gone into annealing. We have started extension, okay? In real-time PCR, you get this weird little thing right here, okay? So remember that synthetic DNA we were talking about a little bit ago? Here it is again, okay? We have a probe that is complementary to some part of the gene that we're expressing, okay? And because it's complementary, it sticks to the DNA. Now, this probe has 2 little pieces on it, 2 molecules. One's called a quencher, and the other one is called a reporter. The reporter, not surprisingly, is a type of green fluorescent protein, okay? So there it is. But this green fluorescent protein cannot fluoresce because this quencher, to quench means to put out, okay? Quench your thirst. You're putting out your thirst. You have a reporter, but that quencher is really, really close, so the quencher is stopping the reporter from fluorescing. But what happens is that as this DNA polymerase walks this way, okay, synthesizing the daughter's DNA strand, when it gets to this complementary sequence, is going to excise it, is going to break it into little pieces. When that happens, this quencher floats away from the reporter, and now the reporter can fluoresce, and the UV light in the thermocycler grabs that light and go, aha, we just synthesized one strand of DNA. Isn't that something? So in real-time PCR, not only is PCR taking place, it is also quantifying, it is enumerating, it is telling you exactly how much DNA is being amplified. Very interesting stuff, okay? So that event is called real-time PCR. Not only amplifying DNA, but being told how much of it is being amplified. Okay, enough for PCR. Now we're going to go into electrophoresis, okay? So in electrophoresis, again, we're looking for a gene of interest. And the first time that we did it, it was using DNA, okay? So what do? Let's go to the picture. Here's a gel. The gel is usually made out of agarose. If you're doing sequencing, which nobody in their right mind does this anymore, but if you're doing sequencing, it would be an acrylamide gel, okay? But we definitely do the agarose gels. The agarose is the same gelling agent as in your Petri dishes, okay? So here's a gel, and then we take DNA, and not surprisingly, we cut it with restriction enzymes to make it nice and short, okay? We load the DNA with a micropipette into the wells, and then we put this into a chamber with an electrolyte. That's a liquid that can pass a electric charge. And we put a positive cathode down here and a negative anode here. And because DNA has a net negative charge, it has a tendency to migrate towards the positive cathode, okay? It does not all move at the same rate. The smaller the DNA fragment, the faster that it moves. The bigger it is, the slower that it moves, okay? The scale is logarithmic. You don't need to know that. So as this migrates out and it resolves, okay, it resolves throughout the agarose gel. And then what you do is you electrophorese this sideways, okay? So you electrophorese it this way in order to resolve the DNA strands. And then you're going to, oh, that's right, our author didn't put that. Then you electrophorese it sideways so that the DNA comes out of the gel and sticks onto a nitrocellulose paper, okay? So now you have the DNA on there, and I gotta tell you, this DNA is invisible to the eye. You can't see it. So what are you gonna use? That's right, DNA probe, right? So now you wash this paper with a DNA probe. What's a DNA probe? Is a complementary sequence of nucleotides that recognizes the sequence in the gene of interest, so it'll stick to it, and it has a reporter like green fluorescent protein. So once you wash it on there and you put it under UV light, the gene of interest, the gene that you're looking for, is going to fluoresce whatever color your reporter is, okay? This event is called a southern blot. Why is it called a southern blot? Because Professor Southern, Dr. Southern, is the one that came up with it, okay? So DNA electrophoresis is a southern blot. Can we do this with RNA? Yes, we can. What do we call it? Well, whoever said that scientists don't have a sense of humor are wrong. We call one running DNA a southern blot after Dr. Southern. One with RNA is called a northern because it's the opposite of southern. And you can do this with protein as well. And with protein, that's called a western blot, okay? So this is ways to cut, resolve, or separate strands of DNA, RNA, or protein and then watch it with a probe and see if the DNA, RNA, or protein of interest happens to be in there. Okay, very neat stuff. All right. Now we're getting DNA microarrays. Same general concept, except that instead of using a gel, now we're going to use a microarray that has wells in it. And each well is going to have a different cell expressing a different DNA or a different part of a DNA. And then, not surprisingly, we're going to use a probe. What's a probe? A probe is a sequence of nucleotides complementary to a sequence of interest with a reporter, okay? So this is what a microarray looks like. It's a whole bunch of wells. Those wells have different pieces of DNA. And what we're doing is we're using multiple probes. So here you can see that some are green, some are yellow, some are red. Some of these are combinations of probes, okay? And what this does is it tells you what DNA or RNA or DNA or RNA is being expressed, okay? So as a DNA microarray, let's see. And really, that's where we're going to end, okay? So have yourselves a wonderful morning, and I'll work on the next lecture on the next chapter in just a second.